I am Dr. Zahid Manzoor. I am working as Assistant Professor in the Department of Pharmacology and Toxicology at the Cholistan University of Veterinary and Animal Sciences, Bahawalpur, Pakistan. We are conducting experiments on the natural products and we are using techniques like necropharmacology for repurposing of the drug. So today we will discuss a recent study which we have conducted on the Phanicum vulgare. So these are the contents. We will discuss first of all hypothesis of the study, then main objectives, introduction, a brief introduction of the study, then a complete methodology, results, and finally discussion. So hypothesis of the study was Phanicum vulgare was having neuroprotective effect because it was traditionally used in neuro disorders, and there are few reports that it is effective against inflammation, and there are also some reports that indicate that this plant has antioxidant activities in different types of diseases. So based on these hypotheses, we designed this study. We have four main objectives of this study. First objective was to identify main phytocompounds, then to identify the pathogenic genes. The third objective was to identify strong ligand protein interactions through molecular docking. And final objective was to predict the possible pathway that is involved in neuroprotection. Alzheimer's disease, as you know, it is a neurodegenerative disorder. The main signs of this disease include memory loss, loss of thinking, and altered behaviors. Pathophysiology is that there is presence of two abnormal proteins in the Alzheimer patients. One protein is the APP, that is called amyloid precursor protein, and second protein it is called MAPT, microtubular associated proteins or tau proteins. So there will be deposition of these two proteins in the neurons and the patient become Alzheimer's. Here is a figure that at the upper side this is a normal hippocampus, and there will be shrinkage in the Alzheimer patients of this hippocampus, as pictures clearly shows that there is a shrinkage of this hippocampus, and there it represents the prevalence, and Pakistan also has the 50% population of Alzheimer diseases. The risk factors of this disease is divided into two main factors. First one is the risk genes that contributes to 60 to 80%. In these genes, these three genes play a significant role: APP, MAPT, PSE, and 1. And whereas 30% lifestyle contributes to the development of this disease. It includes high blood pressure, blood glucose level, smoking status, and the body weight gain. And here is a saying of Dr. Wen Chi that what is good for your heart is good for your head. This is the pathophysiology. Uh, Here is the aggregation of beta amyloid, and these are some of the molecular pathways that are involved. And ultimately, there will be lipid peroxidation and membrane damage, apoptosis, synaptic dysfunction, neuronal death. So what is network pharmacology and molecular docking? Network pharmacology is a computational study that study of the interactions between drugs, targets, and biological systems. Multi-drug interactions is the uh, main target of this network pharmacology. Disease gene drug network and drug repurposing. For drug repurposing, we use this technique. And second one is the molecular docking. It is a computational technique used to predict the interaction between a small molecule ligand and a target biomolecule, typically a protein, enzyme, or nucleic acids. So molecular docking predict binding predictions, interaction analysis, and binding energies. So these two techniques are in silico used, and we can predict the target, we can predict the possible pathways of any phytocompounds. This is a complete methodology. First of all, phytocompounds of F. vulgaris were retrieved from the available datasets, and the second pharmacokinetic analysis of vulgaria compounds were done. Then toxicity assessment of the compounds were predicted. Then estimating of the target of Phenicum vulgaria against ED, and differential gene expression was done. When plot was reconstructed, and finally network pharmacology and cytoscape was used, gene ontology, molecular docking, and finally validation. So these are the some of the significant results. We retrieved 705 compounds from the impaired datasets, 105 compounds from the knapsack. After removal duplicates, we retrieved overall 500 unique phytocompounds. After this, pharmacokinetic analysis were done using MolSoft and Swiss ADMA tools, and these were the criteria that the a compound having good pharmacokinetic must have drug likeness score more than or equal to 0.18, and molecular weight should be less than 500, and Lipinski rules were applied for the pharmacokinetic analysis. These are the 17 compounds and their pharmacokinetic analysis. So 17 compounds were found, evaluated, include for their toxicity analysis, hepatotoxicity, neurotoxicity, carcinogenicity, immunogenicity, mutagenicity, cytotoxicity, and all 17 compounds tested showed non-toxic behavior. So this is a predicted LD50. After that, we plotted the Venn diagram. When volcano plot of the differential genes, we use the GSE122063 dataset that includes 88 brain tissue samples, 44 from Alzheimer's disease AD patients and 40 from healthy patients. Then it was plotted a differential gene expression. Red dots upregulated genes that were linked to mainly inflammation, oxidative stress, and immune responses, whereas blue dots, these all genes were downregulated and it was associated with synaptic mitochondrial function and neuroprotection. And these were some of the blacks, that insignificant genes in black. Then we plotted we found the targets of the genes. AD 6504 genes were involved in the progression of AD, and we get 17 phytocompounds. Then we found there is a relation between these phytocompounds and the genes of the AD, and we found that 143, these are the compound genes or similar genes. So it shows that there is a relation between the phytocompounds and AD. These are the overlapping genes. After that, we constructed the compound target network. As you show, these are the 7 compounds and these are the different genes, and there's a good interactions that this plant has the ability to interact with the genes of this AD. If there will be no interaction, then we suggest that this plant have no affiliation, no interaction with the genes. So a good network, it shows that these compounds have a good interaction with the genes. Then we constructed a PPI network, protein-protein interaction, and among the genes, they have very good interactions. It shows that they can modulate any pharmacological change or pharmacological actions. After that, we found that some of the hub genes from the cytoscape, and these are the top hub genes. Hub genes are those genes which play a significant role in the development or communication of any pharmacological action. We perform the gene ontology enrichment analysis, and in this 169 biological processes were involved, 36 cellular components were involved, and 65 molecular functions were involved. Cake pathway enrichment analysis were also done, and these are the all possible pathways that may be involved in the neuroprotection. Then we validate the results with some of the correlation of these genes APP correlation, 6 hub genes were significantly correlated with APP, and 5 hub genes were correlated with MAPP, whereas 4 hub genes were correlated with PSE and I. These are the correlation analysis. Finally, we performed molecular docking of 3 proteins. These are the 3 phytocompounds which showed best binding energy. These are the binding energies. On the basis of binding energies, we have selected these 3 proteins, 3 phytocompounds, and we selected these 3 proteins. And these are the different interactions. Here is the second protein interaction with the second and third protein. After all, the possible pathways that the Phonicum vulgare phytocompounds, they can modulate PI3K AKT signaling or NF-kappa signaling and pathways, and ultimately they will result cholinesterase inhibition, microglial inhibition, apoptosis reduction. After these leading to antioxidant, anti-inflammatory, and finally we get neuroprotection. So here is the conclusion. The phytocompound also ornithine, cyanurosid, or lutein from Phoenica vulgare showed strong therapeutic potential in the context of Alzheimer's disease. And these are the some of the activating proteins. And it also shows dual mechanism. So finally, we recommend that to conduct in vivo studies to confirm the neuroprotection and to develop optimized delivery systems and assess potential for pharmacovulgare in other neurodegenerative conditions.